going to tell you my story of establishing the genome editing in Nairobi, Kenya. So let me take you a few years back into my journey. 12th July 2004. It was an early morning of a pleasant day in Kampala, Uganda. There was a slight early morning breeze as sun coming up in the horizon. I woke up early and got ready and drove to Uganda to pick my colleague. We were actually planning to visit a farmer in a village near Gayaza. So we were on the road for almost an hour, and then soon we entered into a bumpy Marham road. I looked out from the window of my car and the reality struck me. So before actually coming to the village, I already knew about a problem which the farmers were facing in their banana cultivation. So when I saw out of the window of my car, I saw the wilted banana plants with some barren field. So we parked our car, my colleagues and I, we got out of the car and we looked around. The view was heartbreaking because as far as our eyes can go, we only saw the diseased banana plants wilted and the whole field was like wiped off. And in the corner of that field, we saw a small house. It was a thatched roof house. That was our farmer's house. So we entered into the house, and the farmer was there. He was sitting at the corner of the room. He looked at us with very sad, dull, and depressed eyes. So when we went with a small gift, a bag of sugar for him, and I presented this gift to that farmer. The farmer was almost in tears, and he said, he didn't have even sugar to prepare his tea. So then the farmer stood up, took us out into his field, and he started telling us how his farm was devastated because of this deadly disease. And while he was narrating his story, he was in tears. And after hearing his story, I was in tears. And I had so much of mixed feeling, which I cannot explain in words, but I was very, very sad. On other hand, there was also the desire developed in me to help this farmer. This was a very old farmer, and even, his clothes were very old and teared off, So he explained to us that this field was his sole way of getting food and income to buy things like sugar. So because the field is not there, he had no income. This was not the only farmer who was affected. There were millions of farmers who depend upon banana for their food and livelihood were devastated. So banana xanthomonas wilt, that was the disease which was affecting the farms of the farmers in that village, and also not only in Uganda but also in the neighboring countries. So this disease is a bacterial disease which affects all different kinds of bananas, and it starts with the wilting of the disease and eventually killing the whole plant. The fruits get premature ripe and rotten. So in the end, the farmers cannot get any harvest. Even some of the fruits which looks okay, but if you break those fruits, you will see that they are rotten from inside, so they are uneatable. Once the disease is in the field, there is no other remedy than to cut off the diseased plants and leave the field fallow for some time before the farmers can plant again. So something needs to be done. There was estimated economic losses about 2 to 8 billion US dollar in 1 decade. So you can see how big is the impact of this disease. Not only the farmers who are affected with this disease, they are also the consumers who are getting affected with this disease, because the prices of banana is going up because the production is going down. So you might be wondering, what's my role in it? So my team and I, we started developing the solution to control this disease. And at IITA, we were working very closely with NARO, National Agricultural Research Organization, at Uganda, and we were busy developing the bananas resistant to bacterial disease by bringing the disease resistance gene from sweet pepper to banana. So we tested these improved varieties in the greenhouse, and we got very promising results. We were very excited. Then we tested in between 2010 to 2012, we tested these plants under the confined field trial at the NARO's field in Uganda. And we got very high, enhanced disease resistance under the field condition. So we were very, very excited about that. But it will still take some time before the farmers can get these varieties, because these are the transgenic bananas and you need to follow the national regulatory guidelines before these varieties can be released to the farmer. Then another phase of my life came in, and I was relocated to Nairobi, Kenya in 2011. So I was a bit unhappy to leave Uganda, but my family and I, we packed the bags and we moved to Nairobi. I was a bit sad, but because I have positive attitude, so I also was a bit excited that, okay, so this is an opportunity to get more excitements, new memories in a new country. So after coming to Kenya, my team and me, we created a plant transformation platform. It's only a type in Africa. This is the only one platform in Africa which can be used for developing the genome edited banana, but even the other crops in sub-Saharan Africa. So what is genome editing? Genome editing is a group of technologies which allows scientists to make precise and specific changes in the characteristic of an organism. These type of tools can be used for crop improvement, for example, for making bananas resistant to diseases and pests, and even increasing their yield. So one day in early 2014, I was sitting in my office reading an article on application of CRISPR-Cas9 for plant improvement. And I wondered, can I use this technology for improvement of banana, for making some small changes in banana to make it disease and pest resistance? And as I was going through the article, I was a bit nervous, but also very excited, because all these different type of thinking was coming in my mind. So I was a bit nervous because in those days the genome editing was very much limited actually to the advanced labs in the developed countries, and there was no single report in Africa. So I wondered, why is it not in Africa? Whether there are no resources in Africa, or there is no capacity or training. And I took it as a challenge and made very strong determination that I'm going to establish genome editing platform for the crop improvement in Kenya. And there were many things actually which encouraged me to think in that direction, because we already have the tissue culture transformation platform available. The lab facilities were there. The protocols required for making the editing in bananas were there. and then the reference banana genome sequences were there as well, and then definitely the strong determination was there. So we started working on this. And then to start a genome editing platform, actually the target we chose was the banana streak virus, which is another devastating disease affecting bananas in Africa. So in 2018, we got the breakthrough, and we deactivated the banana streak virus, which was integrated into the host genome of banana using the CRISPR-Cas9 technology. And this was the first successful application of genome editing in banana for the disease resistance. It was the first report globally for any disease resistance using the CRISPR-Cas9 in banana. And so was a happiest day of my life, and many people thought it was unbelievable. The planning for this actually started in 2014. So over the weekend, one day, I was sitting in my house reading an article. So the article I was reading was on human viruses. And after reading that article, actually, the light bulb blew in my mind. And I came up with an idea, very excited. And I went to my husband, and discussed with him, because my husband has background in medical science. So I discussed with him. Later on, I discussed with the banana experts. I also discussed some of my collaborators. And then I came up with a plan after a lot of discussion and brainstorming. And then we started working on this project for deactivating the banana streak virus. And you can imagine, you can feel how excited my entire team was. Not only my team, but even the nation was very excited, because this was the first project in Kenya, in Africa, even at IITA, applying the genome editing for crop improvement. So once this protocol was established for deactivating the virus, it has overcome an hurdle which banana breeders were facing, but also was also the germplasm movement on the continent. But this breakthrough has opened up a path for us to applying this genome editing technology for other diseases as well. And whole the time I was always thinking of the banana xanthomonas wilt, which is also in short called BXW, and the old farmer suffering because of this disease. So as you remember, I said that all different types of bananas are affected by BXW. But there are some wild type of seeded bananas which cannot be consumed because they are seeded. Some of them has the resistance against the banana xanthomonas wilt. And I was always wondering, what is in the wild type banana which makes them resistant to the disease, and that's not present in the other bananas? So this was always puzzling me, but this mystery was solved in 2018, and we got the information that why these wild type bananas has the resistance against this disease. So in 2018, I was actually top of the world, because we had this information from wild type banana, which we can transfer into the cultivated varieties of banana to make them resistant. But we also now know how to edit the bananas. We have the protocol, we have everything available. So I made a plan, and we started experimenting in the lab. I used to spend hours and hours in the lab with my team members, to do all these experimentation. And finally, in 2019, we generated the edited banana plants resistance to BXW disease. And we tested them in the greenhouse. As we were expecting, we got the resistance against the BXW. We demonstrated it in the greenhouse. And that was the excitement moment for everyone. We were so excited that yes, we made it. So the bananas in the edited bananas look exactly the same like the non-edited bananas, because we only made very precise, a small change in one characteristic, that's disease resistance. We didn't touch anything else, we didn't put any foreign gene in it, and that's what the motivation for me to start the genome editing work, because in genome editing work you don't use you don't integrate any foreign gene, you only do very little specific change into the banana's own genome. So the product developed through the genome editing are not considered as genetically modified, they are considered more like the varieties developed through the conventional breeding. I'm so much passionate about this platform. My team has competent scientists, research associates and students, and currently we are using this platform for developing the disease resistant improved varieties of banana, even other crops, and also to train people in order to build a strong capacity in Africa. And we are very much passionate about this platform because this platform was developed for Africans, by Africans, in Africa. You might be wondering, I'm not an African, I am basically an Indian, but by heart I am African because I have spent more than 20 years in Africa. That's where I grow professionally and personally. My 2 kids were grown in Africa. In fact, my son was born in Uganda in 2007. So you can feel it. I am African actually, basically East African. So now we are waiting for the day when this variety, the genome edited BXW resistant variety, will be available to the farmer. So there can be one roadblock for releasing this variety, and that can be the availability of the National Biosafety Guideline for regulating the gene edited product. But the good news there is that Kenya is already developing the guideline for regulating the genome edited product, and I am working very closely with the National Biosafety Authority in their efforts in this direction. So once the gene edited products particularly for the disease resistant varieties of banana will be available to the farmers, millions of farmers, not only in Uganda but in other countries like Kenya, Tanzania, DR Congo, Rwanda, Burundi will be benefited because bacterial wilt is present in all these countries. And I'm still dreaming the day when this old farmer will start growing the disease resistant varieties. That's the time he will get enough food to eat, but also the bananas to sell in the market so that he get money to buy sugar and new clothes for himself. And that day I will say, goal achieved.